So, we will start glycolysis. It has altogether 11 pathway if we consider anaerobic glycolysis too. Otherwise, for aerobic glycolysis it has 10 steps. The step 1 is conversion of glucose to glucose 6-phosphate. Enzyme here is hexokinase. So, as I go on, just start writing from the top to bottom, okay. Don't worry about the structure, see the structure, but write the names only. Write the name of the substrate, product and continue to the next product vertically, one side of the page, not in the middle of the page, one side of the page. Rest of the side you keep it for writing the information regarding the enzymes, okay. So, the first step is glucose to glucose 6-phosphate. Enzyme here is hexokinase or glucokinase. The glucokinase enzyme is present in liver, outside liver, in extrahepatic tissue, hexokinase enzyme is found. Fine. The enzyme transfers one molecule of phosphate from ATP to 6th position of the glucose molecule in presence of magnesium ion. What is the role of magnesium ion? Here it acts as a metal ion and it helps in transfer of gamma phosphate by shielding the charge over beta and gamma phosphate of ATP. The first step of reaction is very important in glycolysis because this step actually traps the glucose inside the cell. The first step of reaction is very important because it traps the glucose inside the cell. Unless this reaction takes place, the glucose which has entered the cell will come out of the cell using different transporters. So, in order to trap glucose, it has to be charged. To make it charged molecule, a phosphate is added to the glucose, so that now it is a charged molecule, so it cannot escape cell membrane. Clear? This is why the first reaction of the glycolysis is first reaction of many glucose utilizing pathways where glucose is needed to be trapped within the cell. Reaction 2, step 2. Step 2 is catalyzed by PHI, phosphohexose isomerase. Glucose 6-phosphate is converted to fructose 6-phosphate and aldose sugar to ketose sugar. Again, this reaction requires magnesium ion. Right? Phosphohexose isomerase. Step 3. Step 3. Catalyzed by PFK1, phosphofructokinase 1. It converts fructose 6-phosphate to fructose 1,6-bisphosphate. It converts fructose 6-phosphate to fructose 1,6-bisphosphate. Why bisphosphate? Why not diphosphate? Because two phosphates are attached to two different carbon atoms. That's why they are called bisphosphate. Mono, di, tri, tetra. Mono, bis, tris, tetrakis. The corresponding to terminologies. Okay. So, bisphosphate means two phosphate groups are attached, one is with C1, another with C6. Now, fructose 6-phosphate thus formed. This particular step is important step, we will come to that later. First, we will complete the pathway. Step number 3. Here also we require one molecule of ATP and magnesium ion. Magnesium ion functions the same way it functions in case of glucokinase or hexokinase. Step 4, it's a splitting reaction. Fructose 1,6-bisphosphate is split into two halves, producing dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, producing dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So, three carbon containing compound are formed from a 6 carbon compound containing compound. It splits the fructose 1,6-bisphosphate from in two halves which forms two interconvertible compounds. The enzyme which splits is called aldolase. It is aldolase A and B both, so you can just say aldolase. Okay. The aldolase enzyme is an example of lyase enzyme. Example of lyase enzyme. Correct? In the next step, step 4, step 5, it is an isomerization reaction, interconversion between glyceraldehyde 3-phosphate, which is in short can be written as GAP, G-A-P, and dihydroxygen phosphate, which is in short is written DHAP. So interconversion between GAP and DAP occurs via an enzyme called triose phosphate isomerase, or in some books they have written phosphotriose isomerase. Whatever you feel, you can write. This particular enzyme is famous because this enzyme is one of the best example of most efficient enzyme in the literature. Most efficient enzyme means the enzyme and substrate go on continuous collision, undergo continuous collision to produce product. So, number of times they form product per 100 time collision gives the efficiency. Okay, so it is around 80%. So it is highest efficiency among all the enzymes. Anyway, step 6. Step 6 is conversion of glyceraldehyde 3-phosphate to 1,3-bisphosphoglyceric acid or 1,3-bisphosphoglycerate. This particular step introduces one molecule of phosphate to glyceraldehyde 3-phosphate, and glyceraldehyde from aldehyde it is oxidized to acid. So it's a step where glyceraldehyde is oxidized as well as phosphorylated at C1 position. So the product is formed is 1,3-bisphosphoglycerate. Correct? 1,3-bisphosphoglycerate or 1,3-BPG. 1,3-BPG. The enzyme here is a little bit longer, long name, glyceraldehyde 3-phosphate dehydrogenase. Glyceraldehyde 3-phosphate dehydrogenase. In short, you might write GAPDH. Glyceraldehyde 3-phosphate dehydrogenase. This particular step, step 6, also require one molecule of NAD, which is converted to NADH plus H plus during the reaction. One molecule of NAD is converted to one molecule of NADH. Now, if you see carefully from step 5 onwards, the stoichiometrically all steps are twice in number. Why? Because glucose fructose 6-phosphate is split into two halves and both the half will show this kind of reaction. So actually this reaction should include two molecules of GAPDH, GAP, converting into two molecules of 1,3-BPG, producing two molecules of NADH plus H plus. You need not to write 2, you just write 1. But remember the reason behind. In the next step, 1,3-BPG is converted to 3-phosphoglyceric acid or 3-PGA. This particular step is step 7. The step 7 is called the first, step 7 is called the first step of substrate-level phosphorylation. The first step of substrate-level phosphorylation. In this step, one molecule of ADP is converted to one molecule of ATP, and that occurs in cytosol. If I say in cytosol, that will not be completely correct because this particular enzyme, what is the enzyme? PGK, phosphoglycerate kinase. Phosphoglycerate kinase. This particular enzyme is located in association with cell membrane. If you can recall, I told you any enzyme where high energy molecules are involved, those enzymes must be membrane bound, otherwise the reaction will not take place. Fine. So this is the first step of substrate level phosphorylation, producing first molecule of ATP. Next enzyme, converting 3-PGA to 2-PGA. Enzyme PGM, phosphoglycerate mutase. Phosphoglycerate mutase. Again, this step require one molecule of magnesium ion as cofactor. Magnesium ion as cofactor. Right? Next step, removal of water from 2-PGA forms 2-phosphoenol pyruvate. Enzyme enolase. Step number 8 or 9. 8. Right? No, 9. Step number 9. 2-phosphoenol pyruvate is converted to pyruvate via enzyme pyruvate kinase. Via enzyme pyruvate kinase. The pyruvate kinase require magnesium ion, and in addition to magnesium ion, they also require potassium ion. This is difference here. They require potassium ion, right? So now this is step 10. Now, if we consider it as an aerobic glycolysis, then step 10 is the last step. Fine. Now, go back to step 1 and you have drawn arrows. So, draw one-sided arrow in step number 1, 3 and 10. Rest all arrows you convert to both sided arrows. 1, step 1, 3 and 10. Make it one-sided arrow. Okay. Now you write irreversible steps of glycolysis 1, 3 and 10. Irreversible steps of glycolysis, step 1, 3 and 10. Energy requiring steps of glycolysis 1 and 3. Energy producing steps in glycolysis, step 7 and step 10. Step 7 and step 10. Okay. Now, rate limiting step of glycolysis 1, 3 and 10. Rate limiting step or regulatory steps of glycolysis 1, 3 and 10. Pacemaker of glycolysis, next point. Pacemaker of glycolysis PFK1, phosphofructokinase 1. Done? Now, the next one, give heading anaerobic glycolysis. Now you write anaerobic glycolysis. Right. The last step. Step number 11, conversion of pyruvate to lactate. Enzyme lactate dehydrogenase. See, again this is both sided arrow. Both side arrow. So it is a reversible step. So only 3 steps are there in glycolysis pathway which are irreversible, and only those steps only we call rate limiting or regulatory step. The major pacemaker step of glycolysis is PFK1, that is step number 3. Fine. Now, regulation of glycolysis, first we will begin with energetics of glycolysis. How do you write energetics of any pathway? Energetics means how many ATPs are produced or how many ATPs are consumed in this particular step. Okay. So like this you write, in left hand side you give heading enzyme, in the right hand side you give heading ATP produced. Left hand side you give heading enzyme and right hand side ATP produced. The first one is number 1. First one is HK or GK, means hexokinase or glucokinase. Here minus 1 ATP. Second step, PFK1 minus 1 ATP. Third step, GAPDH. In GAPDH, in the middle you write plus 2 NADH plus H plus, or 2 NADH also you can write, plus 2 NADH. 1 NADH give rise to 3 ATPs upon oxidation under electron transport chain, roughly. So, we can conclude that it contains 6 ATPs, plus 6. Fourth one, phosphoglycerate kinase, PGK, production of 2 ATP, plus 2 ATP. Fifth one, PK, pyruvate kinase, produces plus 2 ATP. Then draw a line, then add up, net gain, net, N-E-T-G-A-I-N, net gain, plus 8 ATP, okay. Then you give subheading, anaerobic glycolysis. This is aerobic, then you give subheading, anaerobic glycolysis, and continue the table. Continue the same table, but give subheading, anaerobic glycolysis. Step 6, LDH, lactate dehydrogenase. Here, see, minus 2 NADH, this plus 2 NADH, minus 2 NADH, so minus 6 ATP. So 8 minus 6 total plus 2 ATP. So net gain in anaerobic glycolysis 2 ATP. Net gain in aerobic glycolysis 6 ATP, 8 ATP. Net gain in anaerobic glycolysis 2 ATP, correct? Now, I tell you the complicated parts of this particular table. This much you must remember. Then now let me tell you the complicated one. What is the complicated one? Number 1, it has been seen that oxidation of one molecule of NADH does not produce 3 molecules of ATP. It produces rather 2.5 molecules of ATP, right? So 2 ATP, 2 NADH means plus 5 ATP. So put a slash and write plus 5. This is new calculation. This is old calculation. Put a slash, backslash, and then write plus 5 ATP. Okay. Similarly, you do the same thing here, minus 5 ATP. Right? Instead of writing 8, what you write now? Same third way. Plus 5 minus 5. Right? Plus 5 minus 5. Same, 2. Fine? Now, few more complications. Huh? What is that? Now, there are 2 shuttle systems. There are 2 shuttle systems. Not for reaching moon or Mars, but for transport of NADH and FADH2 across the mitochondrial membrane. These NADH and FADH2 are very big molecules. They cannot travel through the mitochondrial membrane. More precisely, inner membrane of mitochondria. Right? So, what do you find here? We need a shuttle system which will transport the electron carried by NADH or FADH2, which are called reducing equivalents. There will be some shuttle, means some vehicle will carry. Shuttle means not shuttlecock. Shuttle means which carries astronauts from Earth to other places. So, this shuttle carries electron from NADH outside mitochondria to inside mitochondria. So, there are 2 shuttle systems. One is malate aspartate shuttle system, another is glycerol 3-phosphate shuttle system. Now, if it undergo aerobic glycolysis, then this NADH must go inside the mitochondria in order to get biological oxidation, correct? For biological oxidation, it has to go inside mitochondria. So, if the electrons are carried by mallet aspartate shuttle, then it does in this manner. In exchange of one NADH outside mitochondria, it produces one NADH inside mitochondria, correct? So, the calculation remains same, no issues. But if the NADH is transported via glycerol 3-phosphate shuttle, then this is somewhat different. It carries one FADH2 in exchange of one NADH2. One FADH2 on oxidation through electron transport chain produces old calculation 2, new calculation 1.5 moles of ATP. Correct? So, 2 NADH, if transported via NADH, a mallet aspartate shuttle, then it will produce 5 ATP. But if 2 NADH are transported via glycerol 3-phosphate shuttle, then it will produce 2 FADH2. That means now it will become plus 3, 1.5 plus 1.5. So, you add plus 3 plus 2, it becomes 5, correct? So, put another slash where you wrote 5 ATPs, there you put another slash and write on top, if transported via glycerol 3-phosphate shuttle. If transported via glycerol 3-phosphate shuttle. If transported via glycerol 3-phosphate shuttle. So, it becomes plus 3, correct? It becomes plus 3. So, that net gain in aerobic glycolysis will be 3 plus 2, 8 became 7, 7 became 5, correct? And in this case, what will be a net gain in anaerobic glycolysis? What will be a net gain here? Same. Anaerobic glycolysis will be same. Why? Because there is no question of production whatsoever by electron transport chain. Fine? Okay. So, now why I am telling all this? Because nowadays they put up questions like this. They will give you 1 molecule of glucose on oxidation through glycolysis will produce 7 molecules of ATP. Explain. They won't tell you just write down, describe energetics. No. Okay. They might ask you in this way. Justify 1 molecule of glucose oxidized form 28 molecules of ATP. How do you find out? This is the way. So, as we will go on describing or see with other pathways, we will go on putting up the calculation to this. Fine. Now, the regulation of glycolysis, when we need glycolysis to be performed, when there is energy deficiency, that is early morning or after the breakfast. When? Early morning. So, what do you feel in early morning? Do you feel early morning after getting up from the bed very energetic and very strong and powerful? You feel more strong and powerful mostly during afternoon, because by that time you have had at least 2 meals. Then you feel most powerful now, actually this time. That is why people play during afternoon, not in the morning, correct? And if you play in the morning, what will happen? You will lose weight. If you play in the afternoon, you will gain weight. If you play in the morning, you will lose weight. It is not about losing weight for an obese person. It is about a sportsman person. For sportsmen also, this is true. Fine. So, when ATP level decreases, this happens. These are the checkpoints. Now, we will see how they are regulated. How these enzymatic steps are regulated. You see, irreversible steps, hexokinase is found in all tissues irrespective of wherever it is located. But glucokinase is a type of isoenzyme, type 4 of isoform of hexokinase, which is found only in liver and which has very high Km value. That means it is more specific toward glucose, but it has less affinity toward glucose. Less affinity means high Km. High Km means more amount of glucose is required to trigger glucokinase active. That means when we have had food, nice, belly full of food, then only when our blood is surged with huge amount of glucose, then only our liver will take up the glucose for future use, will store it. If we are starving, if our glucose level is low, liver glucokinase will never take up glucose from the blood. And if you remember, the liver contains which type of transporter? Got 1, 2, 3, 4. Which type? One, two, all these. 1, 2, all these. So they do not require what? Insulin. So entry of glucose to liver is insulin independent. Now this is something which I don't want you to teach. This will complicate the matter. Forget it. This particular diagram, very often they ask. For the timing it will be difficult to remember, but you just write it down. Let me explain all the pathways. What is it? Fate of glucose 6-phosphate. What is glucose 6-phosphate? The first product of glycolysis. Now it can go to many pathways. Here we have to remember those pathways where it might go. Number 1, see, from glucose obtained from diet or gluconeogenesis. Just write down, glucose obtained from diet or gluconeogenesis is converted to glucose 6-phosphate, right? Glucose 6-phosphate can go to formation of glycogen in liver and muscle. Again, glycogen to glucose 6-phosphate can be converted. This direction is called glycogenesis. This direction is called glycogenolysis. Write down, write down. Fatafat, write down, fatafat, fast, fast. Later on you will forget. Then this is one pathway. So that is synthesis and breakdown of glycogen from glucose 6-phosphate. To and from. Right? Next is glucose 6-phosphate can be exported to blood in the form of glucose. It is converted to glucose, free glucose, and then it can be exported back to blood. When it happens? When there is glycogenolysis, when your body requires more glucose, then liver releases the stored glucose. Right? What next? Glucose 6-phosphate is one of the initial step for hexose monophosphate shunt pathway or HMP shunt pathway, which give rise to a number of different types of sugar phosphate. Ribose 5-phosphate, xylulose 5,4-phosphate, erythrose 4-phosphate, xylulose 5-phosphate, erythrose 4-phosphate, ribose, ribulose, NADPH, all these are produced in HMP shunt pathway. So, the one fate is to HMP shunt pathway or pentose phosphate pathway. Another is, this is glycolysis. production of pyruvate and ATP, correct? Production of pyruvate and ATP. Another step, another cycle which is not mentioned here, write down, glucose 6-phosphate goes to formation of glucuronate. Glucuronate. Glucose 6-phosphate goes to formation of glucuronate, means glucuronic acid, via uronic acid pathway. Via uronic acid pathway. Why they are so important? Because uronic acid pathway produces glucuronic acid, ironic acid, N-acetyl glucosamine, all these products which are found in mucopolysaccharide. Very good. Without this mucopolysaccharide will not be synthesized, and without mucopolysaccharide our stomach, our lungs, nothing will work, our joints, right? Now, regulation of PFK1, this is very often asked. This is the most important regulatory step of glycolysis. How this particular thing is regulated, let me explain. This is very, very complicated step. You have to remember many names. PFK1 convert fructose 6-phosphate to fructose 1,6-bisphosphate. The green color shows the positive modulators. The PFK1, phosphatidylkinase 1, is called an allosteric enzyme. So, the compounds which stimulate allosteric enzyme are not called activators, they are called positive modulators. Different name, positive modulators. And the compounds which inhibit them is called negative modulators. So, AMP, ADP and read out fructose 2,6-bisphosphate are positive modulators. Citrate and ATP are negative modulator. Clear? Now, fructose 2,6-bisphosphate is new to you. Now, write something about fructose 2,6-bisphosphate. In the next para, write down something about fructose 2,6-bisphosphate. How it is formed? It is formed from fructose 6-phosphate. What is the enzyme? PFK2, fructose 1,6-bisphosphate, PFK1, fructose 2,6-bisphosphate, PFK2, same reaction ATP, ADP, PFK2, fructose 2,6-bisphosphate. Regeneration of fructose 6-phosphate via fructose 2,6-bisphosphatase. Fructose 2,6-bisphosphatase. Fructose 2,6-bisphosphatase. Hello? Now, the striking thing is these two enzymes are same molecule. These two enzymes are present in same polypeptide chain. So, enzymes which contain two opposite enzymatic reaction within the same polypeptide chain have got a special name called bifunctional enzymes. Bifunctional enzymes. So, these PFK2 or otherwise fructose 2,6-bisphosphatase, same enzyme, are regulated by insulin and glucagon. Remember, glucagon causes generalized phosphorylation and insulin causes generalized dephosphorylation, just opposite. Glucagon causes generalized phosphorylation, insulin causes generalized dephosphorylation. So, what do you think? In effect of insulin, what will happen to PFK2? Dephosphorylation. Now, what do you think? in effect of insulin, should glycolytic pathway increase or decrease? Insulin glycolysis বাড়াবে না কমাবে? বাড়াবে। কি করে বললে? Sometimes the first answer is correct answer. Insulin, প্রথমে মনে রাখবে কি হবে? Insulin is anabolic hormone. Insulin থাকলে বডির ওজন বাড়ে, না থাকলে বডির ওজন কমে যায়, ডায়াবেটিসের লোক রোগা হয়ে যায়। তাহলে ইনসুলিন যদি অ্যানাবলিক হয়, গ্লাইকোলিসিস এটা অ্যানাবলিক রিয়াকশন না ক্যাটাবলিক রিয়াকশন? Anabolic reaction. So insulin will promote glycolysis. Unless glycolysis take place, glucose cannot be utilized for further purposes. Without glycolysis first step, can you synthesize glycogen? No. So insulin must promote glycolysis. Clear? So insulin causes dephosphorylation. So dephosphorylation of PFK1 will cause deactivation of PFK1. Dephosphorylation of PFK2 will cause activation or deactivation. Insulin must increase fructose 2,6 bisphosphate production. So if you want to increase production of fructose 2,6 bisphosphate, you need to increase or decrease the activity of PFK2. Increase. So dephosphorylation will increase the activity of PFK2. Insulin causes generalized dephosphorylation. So insulin will increase activity of PFK2 via dephosphorylation. More precisely, if you want to still remember furthermore, the phosphorylation takes place near the C terminal of the polypeptide of PFK2, which is removed in presence of insulin. Yeah? So in presence of insulin, there is dephosphorylation of PFK2. PFK2 is activated. Activate PFK2 will produce more and more positive modulator, is more and more fructose 1,6 bisphosphate. So production of fructose 2,6 bisphosphate will increase the production of fructose 2,6 bisphosphate. How? By hormonal regulation, effect of insulin and glucagon. How? By covalent modification, phosphorylation and dephosphorylation. Correct? So what is a covalent modification? What are the different modifications or regulation steps of PFK1? Number 1, allosteric modulators. Name them. Number 2, covalent modification. What is that? Presence of insulin causes dephosphorylation that increases activity of PFK1. That not only increases activity of PFK1, that also increases activity of PFK2, which in turn increases the production of fructose 2,6 bisphosphate, which actually a positive modulator for PFK1. And glucagon does just opposite to insulin. Insulin and glucagon is just like India and Pakistan. We have Arabian Sea on our west, they have Arabian Sea on their east. Correct? Whatever India will do, Pakistan will do opposite. If we look west, they will look east. How? We are always face to face. Right? Similarly. So this is the example of hormonal regulation. Then if you ask covalent modification, then you must mention the phosphorylation part. That dephosphorylated form is active, phosphorylated form is inactive. Clear? This is the flow diagram. Whatever I told is written like this. This particular part is very, very important. If you understand and memorize this part, then I tell you the most important two metabolic path metabolism. One is carbohydrate or glucose metabolism and lipid metabolism for a medical student. Throughout your life, if you have very good understanding of these two metabolism, then you will understand endocrinology like this. All endocrine effects are on this only, either synthesis or breakdown of lipid. Fine, it's done. Okay. Please be fast. Go through it, read, remember, memorize and assimilate the knowledge today only, otherwise tomorrow you will forget. Now this is a very important diagram. You can take a photograph also, later on you can copy. This particular slide is not only important for the regulation of PFK2, but also in case of regulation of gluconeogenesis, which we will teach, we will study later on. Okay. Now, what is this? Same thing. Regulation of pyruvate kinase. It has got many functions. Just remember, it has same action of insulin and glucagon, just like PFK1. Insulin will promote, glucagon will inhibit. Phosphorylated form is inactivated, dephosphorylated form is active. Slight changes are there. Slight changes are there where PK is allosterically activated by fructose 1,6 bisphosphate. Muscle form is actually acted upon by epinephrine. Muscle form is acted upon by epinephrine other than glucagon, and this one is allosterically activated by fructose 1,6 bisphosphate in other tissues. You need not to go this much details. Just remember the same way the PFK1, insulin, whatever effect we have insulin on PFK1, the same effect they have on PK. Now, we studied fate of glucose 6-phosphate. Now we have to study fate of pyruvate, because in the next step we will see the oxidation of pyruvate. So fate of pyruvate, just like pyruvate in the middle, like this. Correct? Then correct? Correct? Cysteine is converted to pyruvate via cysteine desulfhydrase. Pyruvate is converted to acetyl coenzyme. Okay. Pyruvate, acetyl coenzyme has got many access. This will be sufficient. Oh, এটা আছে। দেখো ভুলে গেছি, ল্যাকটেট আর ইথানল।